بسم الله الرحمن الرحيم الحمد لله والصلاة والسلام على رسول الله وعلى آله وصحبه ومن والاه وبعد. Thank you for dialing in, and I do apologize for these few minutes of delay due to a small glitch. So let's get on with this video, which honestly I have no idea how long it's going to be. I am going to try to be brief, but there are many questions and I'll try to answer them as much as I can. And so if you get bored, you can just drop off and rejoin, inshallah. So before getting into this topic, which is of course a serious topic, which is the COVID vaccines and what we should expect, let me just do some disclaimers here. Of course, you know, anything I talk about here is my own personal experience and personal views from what I understand from the data, and anything that is said in this video is only for educational purposes and cannot be or should not be taken as any kind of healthcare or medical advice. That is done with, of course, between yourselves and your healthcare provider. A little bit on my background and why I feel comfortable talking about this topic is basically that I started my career in the early 90s in the FDA. Worked for the Food and Drug Administration for a little bit, and so got to see how things are working on the inside, and I'm proud to say that it's a very robust and very rigorous organization and process for approval of drugs and different therapies. And I, after this brief stay at the FDA, I worked for pharmaceutical companies. So I have worked for various pharmaceutical companies now for the good part of maybe 25, 26 years. So doing mostly early clinical research and clinical pharmacology, which means specializing in early testing of drugs in human beings, and have worked in different disease areas, but mostly in immunology and in infectious diseases. So I have some idea how drugs are developed for this. For these indications, I have worked very, very briefly on a vaccine in my career a few years ago, but this has not been the focus. I'm focused more on drugs for treatment of diseases, but vaccines have, of course, the development of vaccines follows the same principles as the development of other therapeutics, and there are differences, as I will, inshallah, help you understand or present to you, inshallah. I'm as a Muslim, as a person who believes in Allah subhanahu wa ta'ala and that He is really controlling everything. I think what really is happening here is this is what we're witnessing is a fulfillment of a divine promise. So I just have to start by that, saying that the Prophet, peace be upon him, sallallahu alaihi wasallam, said that Allah has never created a disease without creating a cure for it, or except getting old, except this body deteriorating and eventually dying. Short of that, for any disease there is a cure, and this cure can be known by some, not unknown by some, as the text of the various versions of the hadith is, and it's an encouragement to actually do our best to find treatments and to find cures for disease. So let's put that in perspective. Science and faith can never be separated in the mind of someone who believes in God and believes that Allah subhanahu wa ta'ala is in charge. So what is a vaccine? Think of a vaccine as a simulation. When you want to learn how to fly, you go and run or take a flying lessons, and you start by simulation. It looks like the real thing, but it's not. or the fire drills that we have in our schools and our companies. And okay, imagine there is a fire. This is how you will behave. So the vaccine is basically, in quotes, tricking the body so that the immune system thinks that it has been attacked by the virus or by a virus, right? Or an organism, a pathogen. And the body runs through all the processes. The immune system responds to this quote unquote attack, which is again, it's a fake attack, and produces all the necessary ingredients of an immune response, meaning produces antibodies to capture and dispose of the virus or the pathogen, produces cells that are capable of creating the antibodies for a long time, and so on and so on. So it is a simulation, like a drill, as I said. So how do we trick the human body to think it's a virus? That's basically is the principle behind developing different kinds of vaccines, different kinds of modalities. As we hear the mention of like vaccines of attenuated or vaccines of weakened, attenuated or weakened, or vaccines of dead viruses, or viruses are never alive anyway, so like inactivated viruses, and vaccines of mRNA, vaccines with DNA, vaccines with viral vectors, etc. So there's so many modalities, there's so many approaches. But before I get into that, I think a lot of the questions had to do with how do we develop a vaccine? How do we come to accumulate enough data that a vaccine is safe and effective? Basically, here's where vaccine trials are different from drug trials. Because drug trials, you get patients with psoriasis, or patients with rheumatoid arthritis, or patients with a bacterial infection or malaria or anything, and then you actually take a group of patients and you test them with the new drug against, so there's a group of patients that are tested with the new drug, the new therapy, versus the standard of care or placebo, or like nothing. It depends on, of course, if someone has an infection, you cannot just ethically give them a placebo. You have to give them the existing treatment, existing antibiotic versus your new antibiotic, for example, and compare the data, who does better, which group does better, the active drug or the placebo group. And in doing so, you need, of course, you have to go through a process of first, giving the drug to, most likely in the beginning, to healthy volunteers to test their the safety in general, and then going progressively larger in the number of patients and the number of subjects tested, until you get into testing of this drug in typically a few hundred patients, right? Because these are all patients, they do have the disease, right? So, and it takes a few years, from discovery of the drug to testing in animals to testing in early in humans and so on. So the whole process takes, a few years, maybe up to 7 years. It could be as short as, 2-3 years. It really, not 2, sorry, 3 to 5 years. And then, or 5 to 10 years, so on and so forth. So why is the vaccine different? Think of it this way. A vaccine is given to healthy people who have no disease, or not the disease that you're trying to protect them against. And there is a certain incidence rate that happens. So if the incidence rate is 1% in the whole population, then in order to get, not hopefully, because you don't want anybody to get sick, in order to get 100 cases, you have to multiply 100 by 100. So it's like you have to enroll 10,000 people. So 10,000 people in one group, in the active group, and 10,000 people in the in the placebo group or the standard of care group. And that's why vaccine trials, so if you want 200 cases or projected, expect to have 200 cases, you have to do 20 and 20. That's 40,000. And if the incident is 0.1% instead of 1%, then you have to multiply this number by 10. So you can imagine you get into tens or even hundreds of thousands of people participating in these trials. And anyone who is familiar with how costly and how long these trials take, you can imagine and you can, come to understand why vaccine development gets so long and so extensive. The objective of a vaccine is a vaccine trial is to establish, number one, because it's healthy volunteers. So number one, it has to be safe. It has to be, as we call, like non-technically speaking, like squeaky clean in terms of the safety profile, because you're not treating sick patients where you can manage, risk and benefit. Here, you're treating healthy patients where the risk of getting, a particular disease is quite low to begin with, and therefore you have to balance to almost like close to the risk. You have to balance against it almost close to zero. So the other thing, of course, you have to establish is a statistically meaningful result where there is a difference between the active treated group versus the placebo group. So vaccine trials by nature are very large and very long. And also because most of these vaccines are developed by pharmaceutical companies, which are for-profit establishment, and I'm not going to get into, how good is this model of, for-profit drug development and so on. Maybe that's the subject of another video. But for now, let's take it as it is. The reality is these vaccines or any other drug is developed as part of a pharmaceutical company portfolio, which is a business. So you have to make business decisions. You have to manage the three important factors of any project: quality, cost, and time. Cost and quality and time are not always they don't always go, hand in hand. So you have to maintain quality, and this is where, FDA is very adamant, and as well as the European agencies and agencies in Japan and other, developed countries are very strict about how you maintain quality from day one until approval of the drug. Then the timeline, some timeline you cannot control. Like you're running a vaccine trial in a situation where you have, a 1% incidence in the population. You need 20,000 people for the trial. It will take so much to recruit 20,000 people under normal circumstances. So there's parts that you cannot control. The part that you can control is how much money do you put at risk. So for example, if I have a vaccine that or a drug for that matter, that I'm sure, I'm very, very sure this is a very well established mechanism and it's going to work most likely, and safety looks good so far, then I can put more money at risk and start, for example, preparing for the manufacturing of it, even though I have not yet proven it to be safe and effective. And that's the kind of risk that is depending on how confident you are as a pharmaceutical company in that will the vaccine work. And on top, so this is exactly what happened in the case of developing these vaccines, the Pfizer, the Moderna, the AstraZeneca, Johnson & Johnson, and the various vaccines. They put money at risk. And I want to emphasize this. Why did they put money at risk? Because not only are they confident, which can vary from one vaccine to the other, that the vaccine is likely to work, but also because they got a big injection of money from the government. So Pfizer did not get any money from the US government. They got money from the German government because their partner, BioNTech, is a German company. So they got, I don't know how much, maybe the equivalent of $1 billion, give or take. Moderna had received funding from the US government. And so they got this huge injection of money, with the government saying, we believe in you, start preparing your manufacturing, and so we will control the time that we will shrink, and we will compress this timeline as much as possible. Once you show it is safe and effective, the machines will already be producing, or have been producing the drug. So therefore you can get between the FDA, this is something like, I'm not going to say unprecedented, but it's amazing that you have approval, or even the emergency approval of a medicine or a vaccine, and then 2 days later it's hitting the streets, it's hitting the hospitals. This is I think it's pretty much unprecedented. So, and that is because the injection of money in the process by the government. So you should be proud of your, among the other things that you do not approve of and you do not like your taxpayers' money being used for, this is where I think you should be proud of, that your taxpayers' money are actually going to fund this rapid development of the vaccine. The other factor for why, because I think it's a big question, I'm focusing on it a little bit. People are wondering, how come, in the past it took like 5 years, 7 years to develop a vaccine, and this vaccine was like cooked in a year or so, or even less than a year. That's what I'm trying to explain. The other factor which shrunk the timeline significantly is a bit ironic. Because, you look at the incompetent response of our government, of the US government and some other governments in like the UK and others, you have, with this incompetent response, you have more cases per million or per percent. So that's why, you can more easily recruit volunteers, because now the incidence is higher and the incidence of the infection is higher. The people are more motivated to participate in these trials, and therefore you can shrink that timeline, the piece of the timeline I mentioned before that you normally cannot control. But in this case, it's shrunk for you because of the incompetence of the administration in addressing the pandemic. So if you go to a place like New Zealand, which 2 days ago or so, I don't know how many days ago, declared that they have controlled the virus, that the whole island is virus free. It could be foolish to try to go into New Zealand and try to run a vaccine trial. There's no incidence. You need a number that you just cannot work. Or you go to, South Korea or Taiwan or some of the countries where, even Japan, where actually the handling of the virus has been very good. So you don't have enough cases. So you cannot run a vaccine trial in these countries. So, subhanAllah, it just happened. It worked ironically in our favor, that we have more cases. We wish we didn't, of course, but we have more cases, and therefore you can recruit volunteers more quickly, and you have more incidents in the placebo group, and so you can get the drug, the trial moving faster. So what we have seen so far is 2 vaccines, and these 2 vaccines received what is called an emergency use authorization. That is given by a health regulatory authority like the FDA or like the European authority or the UK authority, to have, we have a very pressing need, such as a pandemic or a threat that is imminent, and we have enough information. So there is we have enough information to declare this drug to be safe and effective for the purposes of the situation we are in. Which, if you look at it, you will see that is actually not very different from the final approval. So I don't know how much, to be honest, I don't know how much more work Pfizer or Moderna is going to put into the formal application in order to get the final marketing approval. I don't know. I don't think it's going to be a lot more. I mean, it's probably going to be like longer follow-up for the patients or the volunteers, maybe more assessment about the so-called durability of the immune response, more about the less frequent side effects and so on and so forth. That is expected to happen, but I don't think that the increment between what we know now and what we know at the time of the final approval will be very different. I don't know. So, but I'm speculating it's not going to be very much. So let's get into now what the we hear the term an mRNA vaccine. What does that mean? And people, oh, the mRNA, this is like, oh, ribonucleic acid. It has to do with the nucleus. It's going to go in and mess up my genes and go and alter, and be inherited and passed on and so forth. So, I mean, we have to work through all these terminologies. Vaccines, as I said in the beginning, are a simulation. So they are a kind of like a fake attack on the immune system. So this vaccine can either be the virus itself rendered or made to be non-infectious, in quotes, killed. So it's a dead virus, or actually it's better to call it inactivated virus, because viruses are never alive, actually. They are just genetic codes. Anyway, so inactivated viruses injected into the body. And while these have traditionally been used widely, you have to, in order to produce the inactivated virus, you have to produce the virus itself. You have to produce the real virus first and then inactivate it. So now, to produce the virus, you need what is called the Biosafety Lab Level 3, which is a very high safety level, control, and everybody going in and going out and so on and so forth. So that becomes a challenge in itself. It's not, these BSL 3s are not available everywhere. They're hard to establish. There are a lot of regulations that has to go into them. Into establishing them, and you can also think of the risk of having too many of those. I don't know how much the risk is, but I wouldn't want BSLs to be in everywhere. So anyway, BSL 3s. So this is one, and of course the amount of production of these viruses or in the BSL will be limited to the capacity of the lab itself to produce the virus. So while they have been used with great success in the past, then it's still, scientists and companies have been looking for alternate ways. So I'm not going to go into, there are about 4 or 5 general categories of vaccines, and maybe we can talk about them later. But I just wanted to make the distinction between the inactivated virus, which I just described how it's made, and these messenger or small m RNA vaccines. What is and again, people ask the question, mRNA vaccines, there have never been one that is approved. That is correct. Before Pfizer or Moderna, there have not been an mRNA vaccine on the market. Yes, that is true. However, the research to produce the mRNA vaccination technology has been going on for about 15 years. So in 2005, the effort started, or the publication about them started, And then in the last 10 years, and you could imagine a company like, like BioNTech in Germany, who is, run by these two notable, renowned Turkish scientists. They have been working on, they established the company in 2008 and have been working on this among other efforts, for a long time. So it is not something that just, somebody dreamed up the idea, I'm just going to put some mRNA in an injection and just, give it to people. What is a messenger RNA? A messenger RNA is, by its name, it's a messenger. It's normally produced by, for all the proteins that we make in our bodies, there is a genetic code for them, and the genetic code is maintained in the nucleus, in the small core part of the human cells, and then it's there. And this genetic code, which is consists of the DNA, not getting into all the technical descriptions and terminology, but this DNA actually, that's like a template, and the template produces different kinds of another kind of nucleic acid, which is the RNA. One of the types of RNA is called the messenger RNA. So messenger RNA is manufactured normally in the nucleus. Then it goes out of the nucleus to the protein factories outside. So there's a series, of course, of what happened in for the messenger RNA to. I'm oversimplifying and maybe making things a little bit less technically correct. But think of messenger RNA is normally produced by the DNA, and then it goes to what the so-called protein factories in the cell. And it's an amazing creation by Allah subhanahu wa ta'ala. You cannot help by say, subhanallah, when you look at all this. The messenger RNA goes to the ribosomes. This is the name of these protein factories, and instructs the ribosomes to make proteins. And these proteins can be, building blocks for cells. They can be enzymes. They can be, any kind of factors that we need for our biology and our physiology to be maintained and to be. This actually enables life as we know it. So the messenger RNA comes naturally to produce physiologic proteins. So how can we use this messenger RNA process to actually produce a vaccine? So here's the trick. Here's the how we trick the trick, in a good way, hopefully, in the body to actually produce its own vaccine. If you give the messenger RNA and it does not get into the cells, it's just going to be chopped up and digested and will be eliminated quickly. So we have to give. Let me backtrack a little bit. On the surface of the virus, there are different proteins that cause the virus to gain access to the cell. So now, specifically for COVID-19, there is a protein on the surface of the virus that is called the spike protein. The spike protein binds to a receptor, kind of like lock and key type of situation. It binds to a receptor and goes into the cells. So the receptor, the spike protein is very prominent, very well known, and has been characterized very well by scientists since day one of the virus, or even before. The spike proteins for the MERS virus and the first SARS virus and so on and so forth. So they know how to deal with these molecules. So you manufacture a messenger RNA. And by the way, messenger RNA is a very simple molecule. It's not like a huge complicated molecule. So you don't have to have a biological lab to produce it. So you can synthesize it chemically and then get it into the cell. So the ribosomes will receive, this is a process, and this is where the trick is. You synthesize the messenger RNA, you get it into the cell, and then the messenger RNA will go to these protein factories and tell them to produce the protein that it is coded in this messenger RNA. So the whole, I mean, the idea between or behind this COVID vaccine is we inject messenger RNA that corresponds or that has the template of the spike protein. Give it to the cells. The cells will manufacture the spike protein, and then the spike protein will go outside the cells and get exposed to the cells of the immune system. And the cells of the immune system are going to say, you know, oh my God, there's this foreign protein here. Let's work on it. Let's attack it. Let's produce antibodies and produce cells and produce all the necessary components of the immune system in order to prepare for or in order to defeat this attack. Of course, this protein in itself, A, is not the virus. B, is not infectious. It cannot infect you. It cannot harm you in any known way, at least if it is in your body. And it will produce the same immune response as if the virus is actually present. And that's the whole trick, not the whole trick, that's the principle behind these. So instead of injecting dead virus, you inject or you give the messenger RNA to go to the cell and manufacture the viral protein, and the viral protein now is identified as a threat by the body, and the immune system goes into action and springs into action, and they starts producing these antibodies and immune cells, etc., etc. So a couple of questions. Why don't we inject the protein itself? Okay, people have tried that. The protein itself can be chopped up very easily. It's not going to make it into the cell. Generally, cells have very protective mechanisms. We cannot, just inject a protein and it will go into the cells, or will just go into be taken by the immune system. It's going to be very unstable. It will be chopped up. So sorry, so we inject a protein, it will be digested and will go nowhere. Basically, it will disappear in a matter of hours. If we inject the messenger RNA itself, the messenger RNA also is very, very unstable, and it will be regarded by the immune system as foreign, and there may be an immune response to, immediately inactivated and so on. So what the scientists have been working on for these last, 10 years or so is, a major part, nothing is easy about this, believe me. So it's not, I'm going to say the easy part is the messenger RNA itself. It's all relative. But the most difficult part is, how do you, number one, shield this messenger RNA so it does not get chopped up by the body immediately as is injected? And how do you get the RNA, this messenger RNA, into the cells? And this is where the technology here is called the technology of lipid nanoparticles. The lipid nanoparticles are the capsules. What does that mean? What is lipid? Lipid means fat or oil. Nano means 1 billionth of a meter, so 1 millionth of a millimeter. So that's what nano means, right? The size. Nanometer, just like nanometer or nanometer, is like 1 to the power of 10 to the power of minus 9 of the of a meter. So 1 millionth of a millimeter. That's the size. And particle, because they are particles. So lipid nanoparticles, meaning really, really tiny oil globules. And they're actually not just oil. They are they have the structure that is a structure that is very similar to the cell membranes. The cell membrane in the human body and in animals in general is very protective. It can only pass, things very selectively. But when it sees these lipid nanoparticles, they actually look very similar to the particle, to the cells itself, that they can stick to the cells and then go into, or at least, discharge their cargo, if you will, on the cell surface to go to the cytoplasm or to the cell itself. So lipid nanoparticles are an amazing invention. And indeed, only recently, in 2018, there is a medicine that has been approved by the FDA, which uses the lipid nanoparticles to carry a different kind of RNA, which is called the interference RNA. And this is called Onpattro, Onpattro. It is approved for a rare disease situation, and it is, again, it's by a company, I think it's called Alnylam. And it is Onpattro, Onpattro, and it is a lipid nanoparticle containing an RNA and used to deliver this. So the hardest part of developing the mRNA vaccines is this delivery mechanism. And lipid nanoparticles has so much success now that it's actually the delivery mechanism, the delivery itself is very effective, and when you inject the lipid nanoparticles in the muscle tissue, they actually go very quickly into the cells and release their cargo, when in this case an mRNA for COVID or for spike protein, and then the process of vaccination or immunization begins. This is very, very, very important for the development of the vaccine. So so we can call actually the mRNA we can call it a platform, or we can call it a carrier, because now if we use the mRNA or the lipid nanoparticles, if we use them as a delivery mechanism, we can deliver not only COVID mRNA, we can deliver other mRNAs for vaccination, or we can deliver other types of RNA for different disease situations, and so on and so forth. So it is a very promising technology, and we should all be proud of our scientists actually able to develop something like this. So the advantage of, as I said, the mRNA vaccine over the dead virus is, first, it does not require this kind of safety lab, so you can produce it. You don't even have a biological process you have to have a biological process to produce it. You can actually synthesize this like you synthesize any other chemical. You can synthesize the mRNA. You can also synthesize the lipid nanoparticles, and you can then combine the two and manufacture the vaccine and use it. I hope I didn't get too technical in this, but I thought it was important for people to understand, A, that mRNA technology has been around for a while, for at least a decade, and B, this lipid nanoparticle is a very sophisticated and a very efficient technology to deliver the mRNA cargo to the cells, and that the mRNA is actually going to have the cells, or order the cells to manufacture the antibody, or the manufacture the protein, and then the protein will produce the immune response. Now, so people ask so many questions about what should we expect from this vaccine. And I'm going to go look at your questions here, because I think it's very important that I try to acknowledge and try to answer them, or tell you that I don't know. So big question is, what is the benefit of the vaccine? When obviously the benefit is that we protect ourselves from infection. And because it's really hard to know, we don't know this vaccine, we don't know this virus very well. I cannot say, oh, I'm a very healthy guy or man or woman in this vaccine, this virus is not going to be a big deal for me, and also I'm just willing to take my chances and take my risks. This is really, it's based on what would you say that? Do we have a full evaluation now of who gets very sick and who doesn't? We have some obvious risk factors, pre-existing diseases or pre-existing health conditions, older, advanced age, compromised immune system due to, therapy or, like cancer therapy or other immunosuppressants and so on. But we cannot for sure say that, oh, I'm a healthy guy, nothing's gonna happen to me. That is not very scientific to actually say that. Now, do I take the vaccine to protect others? That's the other big bucket of questions. We read in the news, in the media, that actually the vaccine, people misinterpret actually what is being said. There is no evidence that the vaccine so far, just being vaccinated, will protect or will prevent you, the one who received the vaccine, from transmitting the virus to someone else. And therefore, the medical advice is, if you get vaccinated today, continue to wear your mask, continue to social distance. Don't say, oh, I'm free, and go and hug everybody and be in close proximity to everybody. No, that's bad. So this advice is very sound medical advice. Do not go after you take the vaccine and you assume, oh, I'm protected, I'm not gonna infect anybody, I'm just gonna go and get in in normal to pre-vaccine pre-virus behavior. No. However, and scientists of course have to say that, because scientists go by data. We have to show me the evidence. Okay, the vaccine protects from spreading the virus or prevents spreading the virus. Show me the evidence. Well, the evidence is not there yet. I have to emphasize that. However, when you look at the science, what is your hypothesis? The hypothesis now is for every virus there is a life cycle. The life cycle is first the infection, then the entry into cells, then the replication inside the cells, then the rupture of the cells and the virus going out, then the virus goes out and spreads into the body and goes to every area of the body, including going to the respiratory tract and be in the nasal secretion and be in the cough and in the sneeze and all that. So there has to be some because you get a very tiny amount of virus and then it has to replicate and go there. This may happen with no symptoms. Maybe very little is happening and you are a healthy person and you don't have symptoms, and then the virus is, spread and in your body, and you can cough it and sneeze it, and when you breathe it and so on and so forth. So that's how it happens. There is a life cycle. If you break the life cycle at some point, then you have to assume at least that your hypothesis is going to be, when I break the life cycle of the virus goes in there, and then once it goes into the body, it's met by this army of antibodies, and it stops it in its tracks and takes it and binds it, and it gets eliminated from the body. So if I have to guess what is going to happen, or if I'm going to design a study to explain or to examine what is going to happen, I have to make the hypothesis that eventually, when you have a response, we have a robust response, then you will also be non-infectious. But again, this is underline, this is not proven yet. It hasn't been studied. We should wait for the data to come, but we should be, again, working with the hypothesis that it is a very likely benefit. So if I'm healthy and if I'm thinking of, oh, nothing's going to happen to me, which is, I think it's kind of foolish to say nothing is going to happen to me, or, but I want to protect, my parents and my grandparents and anyone that I interact with, I think it's still a good idea to take the vaccine, because it really highly likely that it will be shown to actually prevent infection. Again, hold it until we see the data, but we're very hopeful that this will be the case, inshallah. When do I start getting protected? So far, for the Pfizer vaccine, for example, you have to take 2 doses. And you have to take 2 doses. This is most likely to get the full immune response. There is an immune response that happens after the first dose. So if you're going to respond to the vaccine, you will respond with a robust immune response to the first dose of the vaccine. However, when you wait for, give it a few weeks and then give it another dose, then you mount even a bigger response. Does that mean you should miss the second dose? No, because the results and the information we have are about based on 2 doses. So get the 2 doses. Do not miss the second dose. So that's one of the questions. After the second dose, then the response is probably going to be maximal in a matter of a week or 2 after the second dose. And then you'll be protected from then on. Now, the question, there are several questions that are being asked here. What are the side effects of the vaccine? The side effects of the vaccine or of any drug, for that matter, are of mainly 2 or maybe 3 kinds. But the 2 major kinds are, number 1, a side effect which we call an exaggerated biological response, or an exaggerated, in case of medicines, drugs, and we say exaggerated pharmacological response. What does that mean? For the vaccine to work, it has to stimulate the immune system. And when it stimulates the immune system is so complex, and there are so many chemicals and cell types and so on and so forth involved. And in part of the response involves the cells releasing some what we call pro-inflammatory cytokines, or think of them as chemicals that cause inflammation. They're large, chemicals that cause inflammation in order to protect the body. This is part of protecting the body. So maybe for some person, it's just gonna be like, it's a little bit of soreness here in my deltoid muscle where I took the vaccine, and maybe a little bit warm around it. I can't move my arm for the rest of the day, and so on. And for another person, it could be fever. It could be feeling like a throbbing headache or whatever, and so on. That is what we call the exaggerated response. Exaggerated response is hard to predict, and it should not stop you. So, for example, if you have a little bit of fever and and headache, and this goes away with Tylenol and so on after the first dose, that should not discourage you from taking the second dose. Of course, talk to your healthcare provider. This is not a medical advice type of video, right? So I can guarantee you that the protein that is produced, or the messenger RNA, or this is not going to give you COVID-19. It will not give you the disease itself. There may be some form of a vaccine which is called the attenuated or weakened virus, that maybe some of the viruses are just like going to wake up, or like not weakened enough that you may still get some symptoms, from the vaccine itself. But this is not the case here with the, I have to emphasize this and underline it. You will not get COVID-19 from the mRNA COVID-19 vaccine. This is an immune response, and this is something that is natural, and actually, if anything, it means the vaccine is working. So provided it's not so severe, it should not stop you, it should not discourage you from continuing with the next dose. Now, the other kind of side effect is called the allergic response. Allergy, in actually the meaning, the literal meaning of the word allergy from Greek, means it works differently. So it works differently in people for some reason that wasn't known, and we know a little bit about it more now. So allergy is in some cases, hopefully very rare cases, hopefully nobody, but it will happen, that you get an allergic response. And the allergic response is an abnormal, unexpected, atypical response based on the nature of your immune system and how your immune system responds to certain allergens. Some people have allergy to dust mites, some people have allergy to pollen, some people have allergy to certain medication. You have to write it in your profile when you go to the doctor. I have allergy to this or that. And then if you have a history of allergy that is severe, to peanuts or to some fruits or to you just make your healthcare provider know about this. Make sure they know this is not, according to the CDC today, actually, yesterday released, just generally having allergy to something. Before that, it doesn't mean you should not take the vaccine. It means you just need to inform your healthcare provider and be ready to treat any kind of allergic response, because it may or may not happen. It's likely not going to happen, but it may happen. You also are allergic to something in this vaccine. But if you know you're allergic to any ingredient of the vaccine, then of course, and these are available publicly, and the doctor will be able to, or the person who gives you the vaccine will be able to advise you. Do not take the vaccine if you're allergic to an ingredient in the vaccine itself. Maybe you can get a different vaccine when it becomes available, inshallah. So these are questions about the side effects. Now, can the allergy be so severe as to go into what we call the anaphylactic shock, where there is or anaphylaxis, where there is a sharp drop in blood pressure, a swelling, and inability to breathe, and so on and so forth? It's, again, these are rare events that we cannot evaluate, but people with a history of this kind of response should be very careful and should really, maybe instead of going to a pharmacy to take the response, to take the vaccine, you may have to take it in a doctor's office where there's, or at a hospital where there is actually the facility to treat that. How long are we going to be immune? Circumstantial evidence only so far that this virus is not very actively mutating. And let me backtrack. The virus, the length of immunity will depend on two main factors: the factor related to the virus itself, and the factors that are related to the person or the human being. The virus itself, if it's mutating very significantly, we don't have an HIV vaccine because the virus mutates so rapidly, and you cannot have a vaccine that is effective against it because the virus keeps changing. Mutate means change. It keeps changing in significant ways. Also, we get the virus, the vaccine for the flu every day because the virus changes very, very rapidly. So that is important to know. So this is one factor, the factor that has to do with the virus. The other factor that has to do with the human, himself or herself is, what kind of immune response do we mount when we have this this virus? What is if the immune response is just going to be some low-level antibodies and a few cells here and there, they are more likely to disappear quickly. But if the immune response is robust, and we have not only the antibodies, but we also have the cells that will produce the antibodies in the future and produce the immune response in the future, then a vaccine can last for a lifetime, or can last for 10 years, or can last for 5 years. These are all data that should be collected. So the answer, I think the short answer is we don't know. But I just wanted to make sure that we understand what determines the length of the response to the vaccine. We have other questions here, and I know this has gone much longer than I expected. Again, feel free to drop off and come back and flip through whatever, but I just wanted to give answer to as many questions as possible. We explained the process and why it takes so long normally, and why it didn't take long this time. What are the worst case side effects? Allahumma afina, may Allah protect you all, is anaphylactic shock. Anaphylactic shock is the worst acute side effects, and if someone has a history of this severe allergic reaction, they should be very careful, and they should actually maybe take the vaccine at a facility where they have all the necessary requirements to deal with the situation. Or talk to their doctor. Maybe they should not be taking the vaccine at all. Funny question here: which is more dangerous, smoking cigarette or taking the vaccine? Oh, you guess, of course, smoking is much worse. What will happen if someone takes the second dose? Oh, and they delay the second dose. I think we should take it into the time that is when studies are designed to investigate certain setups. And we cannot tell you, for example, if you take the doses within a week, or within 2 weeks, or within 6 months. What has been studied is what is being implemented in the clinic. And this is how, Jazakallah khair, address allergy people with weak immune system. Again, we don't know exactly if someone has a weakened immune system, whether they will have enough antibodies and enough response with the vaccine. Basically, it may be that for these people, the best course of therapy will be with the, treatment or prophylaxis. It will be with the monoclonal antibodies that are actually ready-made antibodies to be injected. And these have been approved now, or a couple of them have been approved for treatment, but maybe in the future they will be also studied for prophylaxis. I don't know that. But if someone is unlikely, or someone takes the vaccine and then goes and have an antibody test and there is nothing, then at least they know they're vulnerable, and they know that they may need the monoclonal antibodies to protect them. I don't usually get sick. Okay, I got that. Don't be a macho. Just take the vaccine. What else? How advanced is mRNA science? We said that 15 years, very, very robust. Difference between, okay, we mentioned that. Misconception of side effects. It will not give you the disease. It will give you maybe some side effects related to the immune system itself. Conspiracy theory. No, I don't think anyone is injecting us anyone is in pact with Satan and is going to inject us with chips and turn us into zombies. I don't think so. I intentionally did not attempt to address these conspiracy theories here. I know things, I know how it is in the pharmaceutical companies, and there are no secret chips being involved in any of the process. New strains of virus, that's a very important question. So far this virus has been, viruses always mutate, but so far this virus has been, the mutation does not turn it into a more vicious virus or more or a very different virus so far, but this is all data to be collected later, so we don't know about the strains. Some of these are being the very what do you call the storage condition is a challenge for the Pfizer vaccine, not so much for Moderna, because it is actually for Moderna you can have a this is normal freezing and be thawed and used after that. It's not the -80 or the -70. So that is a what else? Other questions. I'm sorry, getting so long. Do we know how long the immunity will remain? No, we don't. We will learn that as we go. When are we going to be covered? After we get, normally you get the dose, the first dose in a couple of weeks, you'll have antibodies. Then you get the second dose in a couple of weeks, you have more antibodies. So within a month or so, the protection should be in full effect. It could be earlier. Very important. Vaccine will not alter your DNA. So some people have asked, if this is a genetic code, is it going to go into my DNA? The answer is no. This is not a DNA vaccine. This is a messenger RNA vaccine that will work in the outside of the nucleus. It will not get into the nucleus, so it will not go into their DNA. It does not do any kind of the so-called gene editing, which is an amazing approach anyway. This is not going to alter your DNA. It's not going to be like, I'll get the virus and I'll change my DNA and then my kids will inherit it. No. Okay, so alhamdulillah, I think we've covered most of the questions. If there isn't anything else, just send me the question on Facebook, and if need be, may do another video. This has been terribly long, and I do apologize for that. So take your time, watch it in pieces, skip through it and so on, but there may be something that is helpful for you or for us, inshallah. And I hope that things turn out to the best, and we get these vaccines and there's a big success, and then we go over this inshallah very some people think that we should get like 70% vaccinated, 80% vaccinated, and therefore I think we should be advocates for this in terms of, the process is very robust, the process is very well established, the risk is taken, only extra financial risk has been taken, not risking the health of people. I cannot emphasize this more. So please, be confident and just go for it, inshallah, with the advice, of course, of your healthcare provider. And may Allah protect us all. Wassalamu alaikum wa rahmatullahi wa barakatuh.